Before we get started with this lecture, I want to just give you a reminder, and you may want to stop here and jump out and read. Um, you'll remember from our muscle lectures that there was a point where I suggested you read pages. Whether you did or not, I don't know, but I'm sure those of you that did got a lot more out of the lecture having had read them. So I'm going to suggest the same thing. Excuse me, talking about an action potential. An action potential is how the neuron is going to do its job. I strongly recommend that if not stopping this lecture and jumping out and read it and coming back, or certainly before you listen to the next lecture, you're going to want to read pages 444 through 452. Again, 444 through 452 in your textbook deals with what's called an action potential, which is how the neuron is going to do its job. So we finished up last time talking about tracks, okay, that or comparing nerves and tracks, that they were both groups of nerve fibers. Main difference is one, the nerve is in the peripheral nervous system, and tract is in the central nervous system. Now, because, as we mentioned, our neurons can only carry information in one direction, we're going to have two different types of tracks, and they're really quite obvious when you think about it. First, we're going to need an ascending tract. These are going to be spinal tracts that conduct impulses up the cord, and they're going to carry sensory impulses. They're going to carry that sensory information from our receptor cells up through and to our central nervous system. And then if we're going to have ascending tracts, we're going to have descending tracts. They're going to be spinal tracts that carry impulses down the cord and carry motor impulses, impulses coming from the central nervous system out towards the effector organs, those organs that are going to have an effect, those organs that are going to do a job. Next is white matter. White matter is groups of myelinated axons from many neurons. Okay, so when we talk about white matter, question I ask is, why is it white? Don't say because that's the color. Why is the color white? Because it is. It's white. When you look at it, it's going to appear white. But why does it look white? Well, let's think about this. What makes up the myelin sheath? Okay, well, first off, why is a myelinated axon myelinated? Well, because it has a myelin sheath. Well, what makes up the myelin sheath that's going to make it appear white? Well, if we remember, the myelin sheath is layers and layers and layers of the plasma membrane. What's the major component of the plasma membrane? Lipids. Well, why is it going to appear white? Think about a steak. What color on an uncooked steak? What color is that fat on an uncooked steak? Oh, it's white. Look at that. So because of the high lipid content in the plasma membrane, those axons that are myelinated are going to appear white, and we refer to them as white matter. We can contrast that with gray matter. When we look at gray matter, it's going to contain either neuron cell bodies and dendrites or unmyelinated axons. Right, so those are our two possibilities of structures that are going to be within this gray matter. Typically, unmyelinated axons are the exception to the rule. Now, when we talk about gray matter and we identify it in the brain, we refer to it as a nucleus or nuclei if I have many of them. Well, what is a nucleus? It's just like a ganglion, except it's in the central nervous system as opposed to the peripheral nervous system. When we talked about a ganglion before, we said a ganglion was a group of nerve cell bodies, which is what we're saying here, okay? But that ganglion was found in the peripheral nervous system. A nucleus is going to be made up of these same things, these cell bodies and dendrites that are unmyelinated, but they're going to be found in the brain. We can also find gray matter in the spinal cord, and it's found in what we call horns, and that's made up of unmyelinated axons, and it's a very specific area within the spinal cord. Well, now we know the parts of the neuron. We've got some basic vocabulary, so we can just pepper those in. Now, keep in mind, I'm making the assumption that you're going to remember that terminology, so you're going to have to get on that. Now we can move on. What kind of neurons do we have? Well, you know me, I like to classify things by both their structure and their function. So when we look at the structural classification of neurons, we're going to base that classification on how many cytoplasmic sticky outies do they have? How many things do they have sticking off of the cell body? This is the type that you folks are more familiar with, the multipolar neuron. This is the kind of cell, this is the kind of neuron that we had slides for in our tissue lab. We have numerous branching dendrites, right? And if we look, the left of this diagram is our cell body. We have 1, 2, 3, 4, 5 dendrites branching off of that, and only one axon. The axon is coming from left to right, that long strand. This type of neuron, the multipolar neuron, is the most common in the central nervous system. And really because of that, because the highest concentration of neurons is in the central nervous system, it is the most common type of neuron in human beings. Now we have this guy. We've got the cell body in the center, and we have a dendrite end and an axon end. We refer to this as a bipolar neuron, right? Bipolar, 2 poles, 2 ends, and that's what we see. One's going to be a dendrite and one's going to be an axon. Now my question to you is, which one is which? Looking at this screen, is the axon end to your left or to your right? And how do you know? Well, if you look really, really closely to the process to the right, you will see the little gecko toes at the ends of those fibers, at the ends of those strands. Those are our presynaptic terminals. Our axon is the only thing that's going to have a presynaptic terminal, so the right side, the fiber coming off of the right side is the axon, on the left side is the dendrite. One is going to extend from opposite ends of the cell body, and that's why we call it a bipolar, from opposite sides. They tend to be typically rare in the adult body. They are found in some special sense organs. They're really very specific where we're going to find them. In the retina of the eye, in the inner ear, and in your nose. So, excuse me, there's not too terribly many spots where we're going to find these bipolar, but they're incredibly important in order for you to get information about your external environment. Next, we have a unipolar neuron, one pole. And if you see, we have our cell body, and there's only one thing sticking off of it. We have 2 sides to that, but there's only one process coming off of the cell body. These unipolar neurons tend to be very short and divided between a proximal and a distal fiber. The proximal fiber is going to be the left side, which is going to be the dendrite side, quote unquote the dendrite side, and the distal will be the right side, which will be the axon side. And again, I know that because I can see my little gecko toes. I can see my presynaptic terminals. These are found mainly in the peripheral nervous system as sensory neurons. So these neurons are going to be very, very important in bringing information into the central nervous system from either the outside environment or internal environment. Remember, we get a lot of information from our inside, right? Our chemoreceptors, our proprioreceptors, our pressure receptors are all going to use, or mainly going to use, this unipolar neuron. This is a nifty diagram. Okay, it shows us a nice comparison between our multipolar, our bipolar, and our unipolar. The difference is, for the diagrams I used earlier, they kind of went from left to right. These are going from right to left, but nonetheless, all right. I like this for several reasons. It's all 3 on one diagram. The other thing is it's pointing out your presynaptic terminal, so you can see the little kind of nubby ends on the axons and the points on the dendrite side. Right, so that's a pretty helpful diagram. Next, we have our functional classification. And these are pretty broad, and they echo the the functions that we talked about when we started talking about the nervous system. So we're going to have sensory or afferent neurons. These are going to transmit impulses from receptors in the skin, sense organs, muscles, joints, viscera. What did we say viscera was? All of our internal organs, okay? So again, we're saying it's going to take sense organs, skin, outside environment, muscles, joints, viscera, internal environment. We're going to take all of that information, we're going to send that to the central nervous system. They are unipolar except for sensory exceptions, those bipolar exceptions that we talked about. The retina of the eye, the inner ear, and the nose. We have motor or efferent neurons, and this is going to mimic or allow for the motor function of our nervous system. They're going to convey impulses from the central nervous system to the effector organs, those organs that are going to have an effect. They are multipolar, and the cell bodies are within the central nervous system. So the only thing coming out will be those axons that we talked about. They're going to form junctions with their effector organs. Our friend, the neuromuscular junction. All right, so we're going to have our neuron, and we're going to have our muscle. So we're going to have a junction, a connection, a nearness between the neuron and the effector organ. And lastly, we have association neurons, or what is also referred to as interneurons. This allows for that integrative process that we talked about. They're going to lie between motor and sensory information. They're going to be a connector between what's coming in and what's going out. They are multipolar. They're confined to the central nervous system, which makes sense because that's where we're going to need that ability to figure out, okay, here comes the incoming information. What do we want to do with that? What should we do with that? Should we just wait and see if the change gets bigger? Or do we have to do something? Do we have to intervene and make a change to fix whatever's going on? 99% of all neurons in your body are association neurons. These are neurons that are helping to set up networks, set up connections so that information can be spread throughout the nervous system. They allow signals to be shuttled in complex pathways. We keep talking about these pathways, okay? We keep talking about these networks. It's sort of like the electricity in your house. It comes in, but it branches to all of the different rooms, all of the different electrical sockets and things like that. Okay? That's what we're talking about here. We're going to start talking about an action potential or a nerve impulse. Action potential and nerve impulse mean the same thing. This is very, very dense stuff. This is going to give you sort of fits like muscle contraction did. All right. So again, you might want to jump out and read first or listen to this part, read, and then we'll pick up with the really dense stuff in our next lecture. A nerve impulse is a tiny electric current passing along a neuron. We know it's an electrical current. We can measure it. We can use a teeny tiny voltmeter and measure that electricity moving through your body. This is why if you've ever been to physical therapy, they can use an electric stim machine where they put electrodes on your body and cause a muscle to contract from the outside in because we're putting little bits of electricity into your body, just like a nerve impulse. The impulse results from ion movement into and out of the neuron. And that's going to be the problem because there's two different ions, and they're moving in opposite directions. The ions will move across the cell membrane or the plasma membrane through special proteins, those passageways that we talked about way back when. These are ion channels. They are selective to the ions that can pass through. Not just any ion can pass through any ion channel. It's specific to the type of ion we're talking about. What I mean by that is there's only specific ion channels that a calcium ion can go through. Remember our calcium ion release channels? That's one of these, all right? So we're going to be interested in these ion channels because they're going to allow ions to move across